as many of you probably know, I just come back from Cambridge. Thereby, a few people asked me in my lab in Cambridge to send the video of the presentation. Thereby, it's been in English. Another announcement before starting. As I told Eleonora, I would like to reduce a little bit the time for questions and increase a little bit the time for the presentation itself. Because it follows quite a linear path. Thereby, if I skip anything, you wouldn't be able to understand thoroughly the ending.
Yeah, we kind of aimed at 10 minutes.
Yeah, I will say the half an hour, no worries. Alpha-synuclein and the pathogenesis of Parkinson's disease. Firstly, what's Parkinson's disease? Well, it comes in two flavors, actually. There's a sporadic Parkinson's disease and a familial Parkinson's disease. Obviously, the pathology is pretty much the same, but obviously, as you can see from the drawings, the familial Parkinson's disease follows a Mendelian kind of inheritance. So patients have a family history of the pathology. While in sporadic Parkinson's disease, it just happens without a family history. But the symptoms are rather the same, and almost throughout the clinical story of each patient, symptoms normally remain completely motor or almost completely motor. You don't get so much neuropsychiatric symptoms like dementia or whatever. They may happen, but in very late stages of the pathology. The main symptoms are motor, so rigidity, instability, tremor, and actually a very special kind of tremor. Let's say that it just happens when they are resting and not when they are undergoing controlled motion, and bradykinesia, which is low movements. And these symptoms altogether produce quite strange signs like Parkinsonian gait, tendency to festination, so to fall when they lose their equilibrium and fall ahead, and micrographia, this tendency to reduce the dimension of their writing, their handwriting. So, if we collect brains from patients died after Parkinson's disease, what do we get? Well, we get that some areas indeed show quite strong signs of pathology. And the most strongly affected area is embedded in the midbrain or mesencephalon, which is the upper part of the brain stem, bronchoencephalic, the upper part. But not the complete midbrain, just a very special region of the midbrain, the substantia nigra, SM, which is in fact black itself due to a special pigmentations in the neurons of this area. And actually not the complete substantia nigra, actually only the medial ventral part, which is called pars compacta of the substantia nigra, SNPC. So what do we get in the substantia nigra pars compacta of these patients? Well, we get less neurons, as you can see from the drawing. Less neurons, many neurons have died in these patients. And the few surviving neurons show very strong signs of pathology, which consist of very big aggregates located either in the cell body, in which case they are called Levy bodies, or in the dendrites and axons, in which case they are called Levy neurites. Well, so what's the matter? What's special with these cells? Well, they have indeed something special. Well, you know how a neuron looks, what a neuron looks like. Well, we have an input station, which are the dendrites, the dendritic tree, which connect to the cell body, where the nucleus is, where proteins are made, and output station, the axon. The ending of the axon is made up by synapses, which are the communication point between one neuron and the next one. The synapses work by exocytosis of specific molecules, the neurotransmitters, which are perceived by the next neuron, the postsynaptic neuron. Well, what's special with SNPC neuron is that the neurotransmitter is a very special one. It's a catecholamine which is called dopamine. But it's not the only region, although rare, in which you can get dopamine in the brain. There's another quite important region. It's also in the mesencephalon. It's called the VTA, ventral tegmental area, which uses dopamine exactly like the substantia nigra pars compacta. But the VTA is spared by Parkinson's disease. Why? We don't know. Well, we'll have some explanations possibly at the end of the presentation. But let's say that the pathology in Parkinson's disease is not only in the substantia nigra pars compacta, it's almost throughout the brain, but in the substantia nigra pars compacta, it's quite more evident in any other regions. But it spreads, really, it literally spreads throughout the brain. So the next question to be answered was obviously what's inside these living bodies and living neurites. Well, the answer came very late, actually. It came in 1997 when Maria Grazia Spillantini, who was the PI of the lab I had the honor to attend this summer, Professor Spillantini, and she discovered with her colleagues that the main ingredient, the main content of the living bodies and living neurites is a synaptic protein which is called alpha-synuclein. It had been studied in other models, in the electric organ of the torpedo californica. In such model, it had been discovered in synapses and nucleus, thereby synuclein, synaptic and nuclear. But we now know that in mammals it's predominantly synaptic. It's rather not nuclear at all, at least in mammals. It's mostly in the synapses in physiological conditions. In fact, it's very useful in the synapses, we think, because it helps the fusion. You can see the fusion of the vesicles to the membrane of the synapse, because it interacts with the snare proteins, which are proteins that mediate such fusion. So it has possibly this meaning, but it's not only in the synapses, it's also in other parts of the cell. We'll see. The first evidence that synuclein quite matters in Parkinson's disease is the very genetic evidence that a mutation in the alpha-synuclein gene can be causative for Parkinson's disease. Causative means that it has 100% or almost 100% of probability to cause Parkinson's disease. And in fact, both the multiplications and the mutations of the gene can be causative for Parkinson's disease. Multiplication means that there are many copies of the genes, thereby the genes is more expressed, and thereby its tendency to aggregate is increased because you have more copies of the protein itself. And secondly, missense mutations. Which missense mutations? They missense mutations which promote the aggregation. So it seems that aggregation, alpha-synuclein, is very important. It's the key element to get those big aggregates which are called living bodies and living neurites. But the way by which monomers, the physiological, possibly, species of alpha-synuclein aggregate into the big bodies, the living bodies and living neurites, it's not a straight way. It comes through different steps. Which steps? First, oligomers. Oligomers are are small aggregates, let's say 40, 50, this order of magnitude, copies of the protein, of the monomer. And they are soluble. That's a key element, because they're very toxic. We'll see how later. Oligomers further aggregate into big aggregates, which can be called fibrils. Well, the nomenclature is not very clear at all. And aggregates themselves further aggregate into even bigger structures, which are the living bodies and living neurites, those that can be seen at the microscope. So the difference between aggregates and living bodies and living neurites is just the dimension, size. Thereby, living bodies were discovered first. They are really huge in the cell, living bodies. Both aggregates and living bodies and living neurites are insoluble. Well, historically, we first thought that since they are so big, they could impair many physiological functions of cells, and thereby be the key element of the toxicity in Parkinson's disease. Well, this theory was proven wrong. Possibly, it's the very opposite. Possibly, the oligomers, the small species, are toxic elements, and thereby aggregates big stuff. Is just a protective mechanism by which cells just suck oligomers into insoluble structure which precipitates and do not harm the cell anymore. So they may be even a protective element, although they indeed absorb some physiological proteins since they are so big, thereby impairing a little bit the functions of the cell, but that's just a detail. But oligomers are thought now to be the key elements for the toxicity in Parkinson's disease? Well, there are many reasons. One of these reasons, and we'll see how exactly, is that oligomers can catalyze the further oligomerization of the monomers, the physiological species. So if you get many oligomers in one cell, you will get a further increase of the oligomers because monomers will be converted by the oligomer into oligomers. So that's a very first, that's a kind of circle, of pathological circle, vicious circle. And secondly, oligomers can inhibit the degradation of both the monomers and the oligomers themselves. So they inhibit the clearance, thereby you get an accumulation and thereby alpha-synucleinopathy, an accumulation of alpha-synuclein, thereby Parkinson's disease, etc. So what does an oligomer look like? Well, the key element to understand oligomers is that they have a misfolded conformation, misfolded shape. So the first one you see in the upper left is the physiological structure, physiological shape, conformation of alpha-synuclein. It's quite linear and has evident alpha helix structure. Well, this alpha helix structure in the transformation from monomers to oligomers is lost. Thereby you get sort of beta sheet structure. Well, it depends, there are many theories about the conformation of oligomers. This is just one example, obviously. In this example, this hypothesis, you get the oligomers are made up of two parts actually, a core which is insoluble, which is lipophilic, and a shell just around the core, which is made up by the last amino acids. You can see these are the 140 amino acids of alpha-synuclein. You can see that the last 40 are strongly negative, while the first 100 are quite balanced and mostly actually without charge. So quite prone to undergo the packing for obviously due to hydrophobic effect. Well, so you get a shell which is strongly negative, and a core which is mostly uncharged, but if you see, it has a mild positive charge, which is deployed on the very surface of the core. This very mild positive charge is really important because it can interact with phospholipids of the cell membranes, which are obviously negatively charged on the outside surface. And by the way, the tail, which is the negative part, the shell, being a tail, it can easily move. So make new room, new space for the positively charged surface of the core to interact with the membrane. This interaction is followed by a movement, a movement of one of the chains in the core, the lipophilic ones, which is moved into the very center of the membrane. This insertion is a key element to understand Parkinson's disease because it just impairs the impermeability of the cells. So when this happens, when you get oligomers accumulating in cells, the membranes are not impermeable anymore, and small molecules can pass through the membrane. That will have huge effects, as we will see now. So let's start with the microscopic pathology of Parkinson's disease, which I've organized into three big parts: mitochondria, proteostasis, and endoplasmic reticulum. Let's start with mitochondria. Briefly, what's a mitochondria? Well, it's a cell organelle which has many functions. The main function can be said to be producing energy in the matrix, which is the inner space within the mitochondrion. Many catabolic reactions are carried out, and many of these actually flow into the Krebs cycle. And the Krebs cycle produces electrons, well, of course, carried by NADH and whatever. These electrons are then later brought to the electron transport chain in the inner membrane of the mitochondrion, where their flow is converted into energy. Let's skip this. Their flow is converted into energy. What happens if this flow is impaired? For instance, due to some impairment of complex 1, the first of these 4 complexes of the transport chain. Well, you get that these electrons, instead of flowing in the right direction, they will start flowing in the opposite direction. And they will happen to be accepted by other chemical species producing ROS. reactive oxygen species, so oxidative stress, species which are extremely reactive and thereby they cause lots of damage to the cell. Well, it seems that in Parkinson's disease, mitochondria are indeed very impaired, thereby the ROS concentration is very much increased. And proof of the importance of mitochondria in Parkinson's disease can be said to be again something genetic. If we get again the previous table of the causative genes for Parkinson's disease, we get that some of these, quite important ones actually, are mitochondrial related. So that's quite a hint. Another hint, and another very historically important hint, is the fact that the administration of some toxins, mostly pesticides, which we think impair complex 1, are causative, both in mice and in humans, for Parkinson-like syndromes, let's say Parkinsonisms. We'll skip the details from that. But it's important, you see, these molecules bind complex 1 and inhibit its action. Thereby you get more oxidative stress, and in parallel to that, you get Parkinson-like syndrome. Maybe the impairment, the oxidative stress is the very cause of the Parkinson-like syndrome you get, or possibly a consequence. We don't really know. But it's a hint. Mitochondria is important for sure for Parkinson's disease. In fact, we said in Parkinson's disease you get an accumulation of oligomers of alpha-synuclein species, thereby also oligomers. Oligomers can inhibit themselves the electron transport chain. So you see, we get another pathological circle, because the transport chain could be impaired due to Parkinson's disease caused by other factors which we will see, or by the genetic mutations of mitochondrial-related genes, thereby the electron transport chain doesn't work properly. You get more ROS, you get more oxidative stress, and thereby more oligomers. But oligomers also catalyze the increase of the oxidative stress, and thereby the formation of oligomers themselves. It's a circle. And oxidative stress, in fact, can promote oligomerization. How does that work? Well, firstly, it promotes nitration of proteins. This is through obviously the reactive nitrogen species, which are parallel to reactive oxygen species. Nitration happens to protein, and specifically, in our case, to electron transport chain complexes, like the complex 1 we've seen. If those complexes are nitrated, they won't work so properly, they won't work so efficiently. Thereby, you will get an increased oxidative stress, and thereby, again, a catalyzation of the oligomerization, and thereby new oligomers, pathological species. Oxidative stress acts also in another way, which is very interestingly dopamine specific. So when a cell is producing dopamine, oxidative stress can catalyze the formation of some oxidative derivatives, as they are called, of dopamine, like this dopamine orthokinone and dopaminoquine. These derivatives are very dangerous. Some of these have been proven to promote a conformational change in the YEMPS sequence in alpha-synuclein, which is in the chain, which is just a sequence of 5 amino acids in the tail of alpha-synuclein. But importantly, this conformational change promotes the oligomerization of alpha-synuclein, thereby Parkinson's disease. And other oxidative derivatives of dopamine can also make adducts, just bind to alpha-synuclein. This binding also increases the oligomerization. So you see, oxidative stress promotes oligomerization of alpha-synuclein. Secondly, proteostasis. Well, what's proteostasis? It's a neologism. It just means protein homeostasis, and it comes with two systems. One is the ubiquitin proteasome system, UPR. The other one is autophagy lysosome pathway, ALP. Let's see the first one. So the ubiquitin proteasome system is based on the proteasome, which is a huge protein structure, which is in charge for the degradation of several physiological proteins. Every protein has a life cycle. It's produced, it stays in the cell for a certain while, and afterwards it's just degraded. Alpha-synuclein is no exception for this. So what kind of species does the proteasome normally degrade? Well, we've seen that it degrades mostly the monomer or alpha-synuclein. Monomers must be tagged in order to let the cell know that they must be degraded because they are old, they're getting old. What's the tag? Can be two things mainly, ubiquitin and phosphate group. Thereby, in Parkinson's disease, you get that oligomers can inhibit this action, this disruption of alpha-synuclein carried out normally by the proteasome. And thereby, in all alpha-synucleinopathies, you get an accumulation of both phosphorylated and ubiquitinated alpha-synuclein species. So, secondly, the lysosomes. Well, there are several kinds of autophagy. Let's just check the two that are important for Parkinson's disease. Chaperone-mediated autophagy. CMA, chaperone-mediated autophagy, is also responsible for the degradation of the monomer. And it's carried out like this. There's a protein, which is called LAMP2A, on the surface of the membrane of the lysosome, which recognizes certain species of proteins which must be degraded. But in some genetic kinds of Parkinson's disease, this protein is not able anymore to recognize alpha-synuclein. Thereby you get an accumulation of alpha-synuclein, again, an alpha-synucleinopathy. And secondly, macroautophagy. What's this? Well, it's a mechanism by which the cell can degrade organelles which are not working so well anymore. They are old, like old mitochondria, or big aggregates like alpha-synuclein, oligomers, or aggregates, or fibrates, whatever, something big. What happens in Parkinson's disease is that macroautophagy is very much induced but not carried to competition. And thereby you get lots of phagolysosomes with stuff inside which can't be degraded by the lysosome anymore because the lysosome is not working properly anymore. And also this kind of macroautophagy, pathological one, is said to be bad macroautophagy because it's not selective anymore, it just degrades whatever organelle. So even mitochondria, even if they are working very well, they can be recognized by this macroautophagy. You can see that if this happens, you will get a decrease of the number of mitochondria, thereby more oxidative stress, because mitochondria is a key element to keep oxidative stress levels normal. Thirdly, impairment of the endoplasmic reticulum. What's endoplasmic reticulum briefly? It's a machinery of the cell, within the cell, which is in charge of promoting the correct conformation, the native conformation of proteins. And as also a second function, the function of shipping the proteins to their destination. And many proteins just go from the ER to the Golgi apparatus, which is the second drawing, and thereby to whatever destination they may have, for instance, synapses. Well, what happens here is that we get an accumulation of alpha-synuclein and oligomers alongside, within the mitochondria, within the ER. These species, as I told you, have a mutated conformation. You remember the drawing, that it's not the physiological shape of the alpha-synuclein, it's a different shape. And this different shape is perceived by the quality control machinery within the ER, which stimulates the unfolded protein response. The cell is saying to itself, we have misfolded proteins, let's do something, let's degrade them. No, we can't, because the degrading mechanisms are impaired, as I told you before. So the ER is stressed, ER stress, ERS. What's the consequence for this? Well, many consequences actually, but the most important of this is the ER to Golgi transport delay. There's a delay in the transport of proteins from the ER to the Golgi apparatus. And if proteins are quite sensitive to this delay could also bring about a depletion of these proteins at the final destination. And a particularly sensitive protein to this delay is the dopamine transporter, a pump which pumps the dopamine in the synaptic cleft back to the cell, back within the cell. Thereby, if you get less that, you will get also less dopamine, and thereby you will get a synaptic impairment, which could also be the cause of the cell stress and cell death, dead cells. Importantly, you also get a calcium dyshomeostasis. Calcium in cells is normally stays normally in just one place, the ER, there you get the highest concentration of calcium. And it's also highly concentrated outside the cell. But, as I told you, alpha-synuclein oligomers have the property of decreasing the impermeability of membranes. Thereby, in Parkinson's disease, you will get calcium flowing from these two places, ER and outside, back into the cytosol. And the mitochondrion is there to check if we get too much calcium in the cytosol, in which case the mitochondrion starts sucking this calcium within itself, bringing about a mitochondrial dysfunction. And again, oxidative stress, again, oligomerization. So this is briefly the microscopic side. Very briefly, the macroscopic part of PD. Well, this was also revolutionized in 2002 by the very famous studies of Brack and colleagues, which revolutionized the study of Parkinson's disease. Why? Because they chose not to study Parkinson's disease on the brains of patients who have died of it. No, they chose to study it on corpses of people who had died of everything else, who had no history of any neurological condition at all. And what they found out is that they could see some pathology signs like Lewy bodies, or maybe earlier aggregates, also in normal people, in healthy subjects, but with a very peculiar pattern. They saw that the pathology seems to follow a rather strict pathway, which can be said to be caudo-rostral, from the back to the front, caudo-rostral. This pattern becomes symptomatic only when the pathology has destroyed most of the neurons in the substantia nigra pars compacta. So the substantia nigra pars compacta is important in Parkinson's disease because it's the key element in which, whenever you get the pathology there, you will get symptoms, but the pathology had been present in your brain for many years before you get the symptoms. This is a key element. And these studies were brought forward. They could see that there are two places in which the pathology starts, and they are not even in the brain itself. First place is the gut, the enteric nervous system. Second place is the primary neurons of the olfactory epithelium. So Parkinson's disease doesn't start in the brain at all. It starts in neuron, yes, in neurons, but not in the brain, in the gut, at intestinal level, not in the brain. The pathology, so you get oligomers there, you get Lewy bodies, kind of Lewy bodies there, and these pathological species of alpha-synuclein are later carried by the nerves through retrograde transportation back to the brain by the nerves, so by the vagus nerve and by the olfactory. But let's now just two final questions. What is the cause? What initiates Parkinson's disease, and how does it spread? So for the cause, we don't have really any hints actually, because epidemiological evidences are just inadequate. As you can see, they're very, very limited. And surprisingly, even if it starts in the gut, it doesn't correlate with alimentation so much. Pretty much no correlations actually. Although, yeah, of course, there are, as I told you, those kind of toxins, those insecticides, which can easily promote the Parkinson's disease. But I mean, we don't have any evidence that all Parkinson's disease cases are caused by such toxins at all. And secondly, a good hint could come, well, possibly, maybe it's just today's fashion, by a kind of alteration of the species of bacteria present in our gut, because we saw that patients with Parkinson's disease quite have alterations in the ratios of different species of bacteria in the gut. Finally, how does it spread? This is a very interesting evidence. There are many mechanisms. Firstly, this transsynaptic transport, so it could just follow the pattern of neurotransmitters actually. This is the most obvious explanation because we saw from Brack studies that alpha-synuclein doesn't spread throughout the brain following anatomical proximity. No, it follows the connectome, so the connections of structures. If two structures are far away, but they are well connected, the pathology will spread very easily from the first to the second. Well, but that's not the only mechanism. There are a few more. Exocytosis, possibly exosome release, which is something very, very new, and possibly something even newer. There may be some nanotubes made of membrane which connect the cytosols or to neurons. But what's very interesting is that when the oligomers, which remember have an altered structure, get to the second cell, well, they have a prion-like property, so they can attribute their own conformation, the pathological conformation, to the physiologically folded alpha-synuclein monomers, and thereby you will get the pathology also in the second neuron, and there you go, spreading throughout the brain. Just as a brief summary, you get in microscopic pathology, you get three things which are impaired: mitochondria, proteostasis, and DR. Well, that's actually pretty much all the cell, but what's important is that impairing only one of these can bring about the impairment of the three of them, like in a circle, through possibly the toxic action of alpha-synuclein oligomers. Thank you. I would really like to thank Professor Grazia Spillantini for hosting me in her lab in Cambridge for the summer. She, by the way, is also the heir of the Nobel Prize winner Rita Levi-Montalcini. And it was really a great honor and pleasure for me to be in your lab, and must thank Dr. Michela Venezunovic for being such an awesome supervisor for me, and Professor Spillantini, Professor Pizzi, and Professor Pizzarusso Benfenati, who helped me in Italy to study, in order to be prepared for this great initiative.
Kind of understandable at times. I'm sorry, this is just wrong, I guess. There's somebody who's evaluating you actually there?
It's 100% in italiano.
Okay.
Volevo chiederti come cambia la modalità di rottura delle membrane nel passaggio da monomero a oligomero della proteina. Cioè, nel senso, nel primo ovviamente è funzionale al passaggio della dopamina, e invece nel secondo caso lascia perdere i canali?
No, allora, se mi sono espresso male, il monomero non altera la permeabilità di membrana, okay. Il monomero interagisce con le proteine SNER che servono per mediare la fusione della vescicola sianica, ma la membrana è assolutamente intatta. In realtà non sappiamo cosa faccia il monomero, perché in knockout, cioè i modelli animali in cui togliamo questa proteina, hanno alterazioni, cioè non hanno niente di diverso né a livello istologico né a livello fisiologico rispetto ai topi di controllo. Quindi non si sa bene che cosa faccia. Però c'è sempre la scusa alfa-synuclein alfa, perché ce ne sono altre due, beta e gamma, che sono molto simili, per cui
potrebbero soffrire la
esattamente. Prego.
Ci sono differenze tra i pazienti che hanno la moltiplicazione del gene e la delezione del gene?
Ma la delezione in realtà è semplicemente protettiva, la delezione sarebbe protettiva, perché appunto la moltiplicazione semplicemente incrementa il numero di specie, il numero di monomeri, che quindi stocasticamente potrebbero formare un numero eccessivo di oligomeri che supera il numero di oligomeri che la cellula può gestire, per cui a un certo punto interviene la patologia. Quindi la moltiplicazione aumenta molto la probabilità che avvenga questa polimerizzazione. Cioè, in realtà è stata anche proposta, insomma, non so quanto sia possibile di fatto, però è stato anche proposto di bloccare completamente l'alfa-synuclein nei pazienti. Non fa niente, apparentemente, però non sappiamo se nell'uomo non faccio vedere niente.
In che modo?
Infatti, servirebbe una terapia genica con un'efficienza assurda che attualmente non abbiamo. Però ecco, è una strategia terapeutica che è stata proposta. Prego.
È un gene molto conservato nelle specie?
Abbastanza. Sì, anche sì. Sì, anche la struttura è molto simile. Sì.
Quindi viene la mia domanda. Siamo tra le due lottatrici che hanno nel lavoro. Anche l'altra ragazza si chiedeva tante cose, però si può seguire ben scritta. E una cosa che ci chiedevamo è che adesso magari rendo anche più specifica, vedo che sei stato in questo laboratorio e loro di fatto cosa fanno? Cioè qual è il modello sperimentale? Come aggiungono le loro ricerche? Che magari a chi non è nel mestiere può incuriosire. Cioè come si fa a sapere questi fatti, queste informazioni?
Allora, ci sono tantissimi metodi. Allora, ne citerò solo alcuni, quelli più comuni. Allora, un metodo ovviamente è knockout, prendere i topi o comunque l'ingegneria genetica, cioè aggiungere geni, moltiplicare l'alfa-synucleina oppure fare mutazioni dell'alfa-synucleina, tipo la A53T che è la più comune, la più importante. È una mutazione che incrementa appunto la velocità di aggregazione dell'alfa-synucleina. E quindi studiare che differenze ci sono, se i mitocondri sono più stressati, se c'è accumulo di qualche cosa, depressione di qualche proteina. Oppure un altro metodo potrebbe essere, come vi accennavo, l'utilizzo di pesticidi, di quel paraquat, NPTP e compagnia, che danno luogo a queste sindromi simili alla malattia di Parkinson nei modelli. E una volta ottenuto un modello buono, cosa molto difficile, bisogna poi studiarlo e a questo proposito si possono usare tutte le tecniche dell'arsenale della biologia molecolare. Principalmente si usano gli staining, quindi l'immunostaining nella fattispecie, cioè anticorpi marcati in qualche modo, colorati o in qualche modo, insomma, marcati, ritrovabili dopo la procedura di colorazione istologica, selettivi per qualche tipo di specie, anche se hanno i loro problemi, perché gli anticorpi intanto non sono mai completamente specifici, quindi c'è sempre del background e di solito è anche abbastanza importante. E poi sono roba grossa e roba polare, i corpi di Levi sono roba apolare e cioè non è sempre fattibile utilizzare gli anticorpi per legare le strutture grosse sull'alfa-synucleina, per cui una maniera per ovviare questo problema potrebbe essere utilizzare anziché degli anticorpi direttamente dei fluorofori, che sono roba piccola e roba polare, perfetta per andare dentro ai corpi di Levi o agli aggregati, e poi ovviamente con l'immunofluorescenza, cioè con il microscopio fluorescenza li andiamo a ritrovare. E poi le tecniche sono tantissime. Ecco, un'altra cosa importante di Western blot è la separazione sequenziale, cioè è importante riuscire a distinguere le componenti, diciamo, solubili da quelle insolubili. Per farlo si prendono i tessuti di modello urino o anche di un paziente post-mortem, si omogenizzano, cioè vengono letteralmente frantumati, resi liquidi, dopodiché si dissolvono in una serie di mezzi, diciamo detergenti con potenza crescente, e dopodiché si prendono le singole frazioni solute dai vari detergenti e si fa un Western blot, cioè si separano in base alla dimensione le proteine contenute. Si aggiunge un anticorpo per l'alfa-synucleina sul blot e si cerca le bande dove c'è l'alfa-synucleina, cioè a quale peso molecolare vediamo queste specie. E con questa metodica possiamo vedere se le specie che troviamo sono grosse o piccole, se sono più monomero o più oligomero, ad esempio. Quindi ci sono varie metodiche.
I pesticidi, una domanda più piccola, che tu sappia, costano tanto, è una via costosa oppure è facilmente reperibile, tipo il paraquat?
No, non è costosa, però non sappiamo bene cosa fanno in realtà. Allora, ad esempio, l'MPTP non passa la barriera ematoencefalica, però, ecco qui c'è anche qualche dubbio, è riconosciuto dal DAT, dal trasportatore della
Io
dopamina.
sapevo questo, sì.
Per cui questo potrebbe essere un possibile bias, perché di fatto è tossico, però se lo prendono solo le cellule dopaminergiche, per cui magari per quello che dà una sindrome simile al Parkinson, quindi quello potrebbe essere un fattore negativo, ecco, che va un po' a scapito della tesi. Però ce ne sono anche altri. Il paraquat, ad esempio, passa la barriera ematoencefalica e sembra che anche quello limiti il complesso 1. Poi non è detto, ci sono 1000 articoli che dicono che non fa niente al complesso 1.
Infatti l'hai scritto
si legge di tutto. Il punto è che sull'aspetto mitocondriale della malattia di Parkinson si legge tutto il contrario di tutto, davvero, e gli articoli sono davvero contraddittori fra di loro. Senz'altro il mitocondrio è coinvolto, e probabilmente è molto coinvolto, però potrebbe essere semplicemente una maniera di entrare in questo circolo vizioso, una delle varie maniere. Una volta entrati nel circolo si ha l'affaticamento e la disfunzione di tutte e tre le componenti che ho schematizzato.